Hi, I'm Anand, and I'm going to be talking about DNA gyrase. So the first thing I want to talk about is the topology of DNA itself. And so that's actually like, how does DNA exist in 3D space? And so although we might normally think of DNA as existing as circular or linear, it turns out that in nature, DNA actually exists sort of coiled up around its own superhelical axis. And so there's actually a lot of natural supercoiling. And so one of the major reasons for this is because during replication, when enzymes like helicase pull apart these two strands of DNA, this causes a lot of tension ahead of the replication fork. And so this results in some positive supercoiling. So there exists this whole class of enzymes called topoisomerases, which are basically meant to regulate this over and underwinding and sort of get rid of knots or tangles that might occur during these processes. And so here's some examples, but I won't get into them. So I want to focus on DNA gyrase, which is a unique topoisomerase, and that it's the only topoisomerase we know of to date that actually introduces supercoiling instead of allowing DNA to relax and getting rid of supercoiling. And so the reason that this exists is because DNA gyrase can work ahead of the replication fork and basically introduce some negative supercoiling, and then helicase will come and cause some positive supercoiling, and hopefully this will balance out. And so on the right over here, we can see that DNA, which is being acted on by DNA gyrase, you can see the effect of DNA gyrase over time because at first, this DNA, which is relaxed, takes a long time to get through this gel. And as it becomes more supercoiled, as DNA gyrase works on it, it becomes quicker at moving through the gel. Okay, so let's explore the mechanism and structure of DNA gyrase to sort of see how it actually does what it's doing. So the first thing it does is it grabs a section of DNA, call this the G segment. Then it grabs another section of DNA from the same molecule. We can call this the T segment. It breaks open the G segment, passes the T segment through this breakage, and this basically introduces extra supercoiling. And so here on this right, we can see a model of, or the structure of like how this is actually this bottom dimer is actually breaking apart the G segment of DNA. And so, yeah, you can see that it's applying tension to these hydrogen bonds and it'll eventually split it up. And then another part of the molecule will do the remainder of the work. Um, the last thing I want to talk about is some of the anti-cancer treatments that are being focused on DNA gyrase. And so basically, there are a bunch of small molecules that can stabilize the DNA-bound state of DNA gyrase, immobilizing it, making it ineffective. And the reason that's a novel cancer treatment is because cancer molecules, cancer cells have a lot of topoisomerases because they're replicating really frequently. And so these poisons are much more effective against cancer cells than they are against normal cells. And so that's why there's a lot of research in that area. Thanks so much. Yeah.